Bismillahirrahmanirrahim. Welcome again, guys. Welcome to another video lecture by Kami Biology. In this video lecture, we are going to talk about the differential screening. So we are actually going to talk about the screening. So we are actually talked about the screening techniques in our previous video lecture. So we already talked three screening techniques in our previous video lecture. If you guys missed it, that video lecture, it is present into my channel. You can watch it, and it is actually the series of video lecture which is going on. And in this video lecture, we are going to talk about differential screening technique, through which we can identify the colony or the bacterial cell having or the bacteria or the host cell having our recombinant DNA vector. Okay, so let me clean the whiteboard and let me start. Okay, so let me write differential or plus minus screening. Okay, now what actually we are going to do in this topic, in this screening. In this screening, we will make a probe in oligo DT column. Oligo DT column is actually a column in which a messenger RNA is always present, and according to this messenger RNA, we will load it our messenger RNA which is present in the colony or in the genetic library. We will load it each messenger RNA, and then the complementary are made, and then we will separate the each of the cDNA or each of the mRNA, and then we will make the complementary of this sequences, and then this complementary sequences which we make from this oligo DT mRNA, then we use this one cDNA as a probe. The cDNA having also radioactive substance, we use it as a probe, and we apply it on our means on the nylon filter paper, and then we will check it that which one probe are bind on the colony. Then we will see it under the X-ray film, and we will make sure if there is any detection are found on X-ray film, then we will make sure that actually this one colony have all those host cell that can carry the recombinant DNA vector. Now how we can do it, let me explain. For this purpose, what actually we have already the plate. This is, let's suppose, the plate. In this media, this is neutron agar plate. In this media we have the colony, as you can see. Okay, now this we will make two plate according to this plate. So we will make two copy of the plate, as you can see. So we will grow the same cell onto the next two plate. These media are actually the differential media. Means in that media we can grow any type of the bacterial cell or any of the cell. Now that's why we can call that differential media, and the technique is known as differential screening. Now what actually we are going to do in this case? In this case, we will leave this plate as it is, but this one plate we will expose it, or we will pour some serum, some solution of the serum in this plate. So let's suppose we will add some serum. Now after the addition of the serum, the plate, inside this plate there is some other colonies are grow. Each colony have special kinds of the mRNA, but we don't know the exact mRNA, and we don't know which one colony are growing in this plate. But there are some other colonies are also grow, or there are some other bacterial cells are also grow. Some other cells are also grow in this plate. So let's suppose we have the same plate as you can see, but in this case we get, and we also get some other colony as you can see. Okay, now these are the new developed cell, and these developed cell are formed by this addition of the serum. After that, what actually we are going to do, we will extract the messenger RNA from each cell. So let's say these are the messenger RNA as you can see. These are the other messenger RNA. Now these are the messenger RNA of the developed cell, and these are the messenger RNA of those cells which are present into this master plate, into that plate. After that, what actually we are going to do, we will put all these messenger RNA into oligonucleotide, into oligo-DT, into oligo-DT column. So let's say this is the oligo-DT column. So inside this oligo-DT column, there is already the messenger RNA which is present as you can see. Let me draw this messenger with this blue color. Okay, and here we also loaded our own messenger RNA of the cell. So it will make the pair as you can see. So when it will make the pair, so one of the there is no one or single messenger RNA. A lot of single messenger RNA are present in this oligo-DT column. This is what this is actually, oligo-DT column. And we have not only one single messenger RNA which is present in this column. A lot of messenger RNA are present, and these are a lot of messengers RNA which we extracted from this master plate, and then we loaded it into the tube, and then we get the complementary of each. And after that, what actually we are going to do, we will separate it from this oligo-DT column, and then let's suppose we separate the messenger RNA from this oligo-DT. Let's suppose these are the messenger RNA as you can see. Now after that, we will loaded the reverse transcriptase enzyme in the presence of in the presence of 32p dinucleotide triphosphate. So let's suppose this is the reverse transcriptase enzyme plus having all the, what we can say, 32p dinucleotide triphosphate. Okay, and then with the help of this reverse transcriptase enzyme, we will get the complementary of this mRNA as you can see. Now we will get this complementary, and after that we take this complementary mRNA with this P32 as you can see. So this is P32. Now this is the radioactive substance. After that, we will separate it because this is an unstable form. We will separate this. This is no oligonucleotide. This is actually oligo probe. let's suppose this is the probe. Now we use this probe and we will apply it on the plate, and then we will check the detection, the coloration, the fluorescence on the plate. Now what actually we are going to do, now our probe are made, then what actually we are going to do, we will put the nylon filter paper onto this surface. Let's suppose this is the nylon filter paper. Onto the nylon filter paper, what actually we have, we have all the cells, as you can see. This is called replica, and after that the same procedure will also perform with this technique. We treat the nylon filter paper with SDS solution, and after that the structure is changed. Let me draw. Now this structure of the colony shows that all the bacterial cells are ruptured and we have just the genome of the bacterial cell. Then we put this filter paper onto the solid surface having temperature 80 centigrade, or we expose it with the UV light to fix all the colony. After that what we are going to do, we add our probe. So then we add all these probes. Okay, so if the probes makes the complementary with this any one of the colony, let's suppose this one colony, this one colony, and this one colony, if they make the probes means the complementary RF matched or formed, then any one of the colonies shows some fluorescence onto the X-ray film. Then we will see the final result on X-ray film. So let's suppose this blue, now this blue one rectangle can represent the square can represent the X-ray film. Okay, this one colony don't have any characteristic, this one colony don't have, but let's suppose this one colony shows some of the characteristics, and they have also show some of the characteristics, and the rest of the cell we have also the same, as you can see. Now once we get the spouts onto the filter paper, then we make sure that actually these spouts are these two colonies, and then we will check these two colonies onto the master plate, and we will make sure that actually these two colony have all those bacterial cell or all those vectors that can carry our target DNA, and then we will use it for our purposes, different kind of purpose. We are using this kind of vector. So the same procedure we also perform with this plate. We will also put the nylon filter paper, and we make a replica onto the nylon filter paper, and then we wash the nylon filter paper with SDS page, and then we expose this nylon filter paper onto 80 centigrade, or we put it or expose it under the UV light, and then to fix all the colony, and then we will place this nylon filter paper under the X-ray film. And after that, on the X-ray film, we will check some spouts. Remember, in this case, we will detect none of the spout, because we also loaded the antibody here as well. Okay, so we also loaded our we also loaded the probe here in this case. But let's suppose this is also another filter X-ray film. So in this case, there is colony present. We can detect the colony, as you can see, but let me draw. But here there is no colony. In this case, colonies are present, but these two colonies are not present. It can show that messenger RNA not binded here. Okay, and these two colony have actually those means in between in each colony there are all those bacterial cell or all those vector which can carry the target DNA. So this is called the differential or plus minus screening. Okay, I hope you get idea about the differential screening. Inshallah, in the next video lecture we are going to talk about blue-white screening. Thank you so much for watching this video.